So their aims of the PhD projects of the students was to find selective inhibitors of carbonic anhydrase 9 and 12 who are involved in the pathophysiology of cancer. And the request was based on molecular modeling studies, and those I'm going to present right now. First, I'm going to start with discussing the importance of human carbonic anhydrases in the pathophysiology of cancer. So basically, all carbonic anhydrases perform a very simple but still very crucial reaction, which is the reversible hydration of carbon dioxide, as you can see over here. And this results in the in the production of bicarbonate and acid ions. So basically, bicarbonate is involved in carbon fixation, and this bicarbonate can be used in the biosynthesis of different compounds. This is a very first important product. The second is, of course, that bicarbonate, which is an ion, can influence the membrane potential and thereby have impact on the action potential. And also, as you can see over here, it is involved in the control of hydrogen ions, concentration of hydrogen ions. So therefore, it's also involved in the regulation of pH in many kinds of organisms. So there are different kinds of carbonic anhydrase enzymes, as you can see over here. These are structurally very different metallo enzymes, so they contain different metal ions, which are important, which are an important part of the active site, and they actively take a role in the catalytic activity of these enzymes. And these enzymes, even though they perform exactly the same reaction, they have very different structures and physiology, especially structures, I'm sorry. So these are basically, these are dimers. This can also be dimers, but here the binding pocket is located at the interface at the dimer, while here it is in the middle of the enzyme. And as such, you have different kinds of structurally related structurally unrelated enzymes that perform the same reaction. So most likely, due to convergent evolution, different kinds of enzymes have learned to pick up the same reaction. So they are structurally very different, but they perform the same reaction. We are mainly interested in alpha carbonic anhydrases. They are present in humans, and there are many different forms. For example, you have the cytosolic ones. These are the carbonic anhydrases 1, 2, 3, 7, and 13. I have indicated 1 and 2 in red because these are very abundant inside different tissues also. So it has a very wide expression profile. Carbonic anhydrase 5A and 5B are mitochondrial types. We have secreted carbonic anhydrases and membrane-associated carbonic anhydrases, which have their active site on the extracellular side of the membrane. They are membrane-bound, either through a transmembrane domain like these three enzymes or via lipid molecules. And I have indicated carbonic anhydrase 9 because they are related, they are involved in the pathophysiology of cancer. So in green are the target carbonic anhydrase that we want to target with our selective inhibitors. In red are the off targets. They are widely distributed in the cytology, but we do not want to target them if we want to target cancer cells. So therefore, these in red, these in green, and then there are 3 carbonic anhydrases without the zinc ion in the active site, without any metal ions in the active site. So therefore, they lack their catalytic activity, and these can be seen over here. So if we superpose all the structures of the alpha carbonic anhydrases, the human ones, the non-human ones, 1, 2, 9, 12, whatever, if you superpose them, you will see that they have very similar backbone fault. The ion, the zinc ion, is located in more or less the middle of the enzyme, but the backbone fault is very similar. There are some differences, of course, because these systems are not static. They are flexible. They undergo Brownian motion, of course. Therefore, there's some conformational freedom is present, but still, if you look at the general structure, you will see that they have a recognizable three-dimensional structure, as you can see over here. If you look at the amino acid level, then the overall sequence similarity is reasonable, as you can see over here. Especially we're interested in the sequence similarity between the off targets 1 and 2 and the tumor-associated 9 and 12. And there you see that you have between 30 and 35% overall sequence similarity. But the problem is this, if you look at the active site, then you will see that the similarity is much higher. I'm trying to get to the next screen. Then you see that the sequence similarity is much higher. If I remember correctly, it's above 80% or so. Still, there are important regions that might be used to target those specific regions. And by targeting these specific amino acids, possibilities exist to develop selective carbonic anhydrase inhibitors. And I have highlighted them or put them in red boxes, as you can see over here. Let's start with this one. This is in the threonine 200 in almost all carbonic anhydrases. However, in carbonic anhydrase 1, there's an histidine very close to this zinc ion, which is part of the catalytic site. And because of steric bumps, you can get some degree, not too much, but some degree of selectivity. So this is one that you can use. There are more important ones. For example, this phenylalanine 91% in 1, and this phenylalanine 131% in carbonic anhydrase 2. And also you see that asparagine, glutamine, lysine, these are significant changes in the active site that you can target with ligands. For example, this has a positive charge. These two have hydrogen bonding acceptor and donor capacities. Here you see either pi-pi stacking possibilities of our hydrophobic interactions. So this gives you some kind of starting point that you can embed in your virtual screenings to obtain selectivity, even though it's not too easy, of course. So if we look at more detail into the active site, here you see the overall structure of the carbonic anhydrase enzyme. The zinc ion, which is part of the catalytic site, is located here, bound by 3 histidines, and these are conserved amongst all alpha carbonic anhydrases. And you see that you have an hydrophobic and hydrophilic part of the active site, and basically that has to do something with its physiological role, of course, which I want to discuss here. So here is the zinc ion bound by these 3 histidines, these conserved 3 histidines. So in the resting state, a water molecule enters the active sites, binds to this zinc ion, and then very close to this zinc, there's a histidine 64, as you can see over here. It's known as a proton shuttle. It's conserved amongst many, if not all, alpha carbonic anhydrases. And this one plays a role in shuttle away 1 hydrogen from the water molecule to the solvent. So by doing this, by performing this reaction, it creates this ion. So what happens next is that carbon dioxide enters the active site using the hydrophobic parts. It binds very close to this ion, and a reaction occurs to form this ion. And then this dissociates away using the hydrophilic part in the active site and goes to the solvent. And by removing of this ion, the zinc is empty again, and a new water molecule can enter or bind, and then a new catalytic cycle is possible. The reaction more or less occurring in all carbonic anhydrases. The ion may be different, the histidines may be different, but the reaction is the same and the result is the same, of course. So if we go back to alpha carbonic anhydrases, then inhibitors can do different things. They can form an interaction with the water molecule bound to the zinc, or they can form an interaction with zinc. So instead of the water, the inhibitor is bound to the zinc, or they can be located somewhere else in the active site, preventing this reaction from happening, or they can enter an allosteric site, changing the conformations and the dynamics of the active site, and therefore influencing the rate of the reaction. In our work, we have been focusing on inhibitors that directly bind to these catalytic active zinc ions. Okay, so now the physiology of carbonic anhydrases in the pathophysiology of cancer. So as you most likely all know, in healthy cells and differentiated tissues, you have two main metabolic routes in the presence of oxygen, and when you have hypoxic conditions, normally you have the reaction oxidative phosphorylation, where glucose is metabolized according to this pathway, and it gives you 36 moles of ATP per mole of glucose. And under, well, when you have not sufficient oxygen around, you have anaerobic glycolysis. However, if you look at cancer cells or proliferative tissue, then in hypoxic conditions, but also when oxygen is present, but mainly in hypoxic conditions, this reaction mainly occurs. It's known as the Warburg effect. And because of this, a lot of acid is basically generated. So this pathway consumes a lot of glucose, and therefore results in a lot of production of a lot of acid inside the cells, which is of course not what the cell prefers. So the cancer cell, or cells in general, have a protection mechanisms, and cancer cell, it works like this. Inside the cell, inside the cytosol, you have these carbonic anhydrase 2. This is a very it's very common amongst many different kinds of cells. It's widely distributed, and this carbonic anhydrase 2 performs following reaction: decarbonate, and the acid, the hydrogen ions, are combined to produce water and carbon dioxide. And this carbon dioxide, by means of passive diffusion, can pass through the cell membranes and go out of the cell. There you have carbonic anhydrase 9 and also carbonic anhydrase 12. And these enzymes then combine this carbon dioxide with water molecules to produce bicarbonate and hydrogen ions. And then the bicarbonate, using many or different mechanisms, is then produced back again inside the cell, where it is again bound to hydrogen ions to again form carbon dioxide. So basically you have a carrying system, which is carbon dioxide bicarbonate, that carries hydrogen ions from the inside of the cell to the outside of the cell. And because of this, the pH values inside the cytosol goes to normal physiological values, while on the outside acidification occurs. And this acidification helps different kinds of metalloenzymes on the outside that play a role in metastasis and other kinds of, I would say, specifics of cancer cells. So this acidic environment is important for the cancer cell to perform its tasks, and also gives it an evolutionary advantage over the healthy cells, which are more sensitive to acid compared to this one, because these carbonic anhydrases also can perform the reverse reaction. If it becomes too acidic, they can neutralize it somewhat. So they have a control of their pH, intracellular and extracellular pH values. So now let's have a look at these carbonic anhydrases 9 and 12, which are very important in the protection of these cancer cells against acidification. The control mechanisms is influenced by hypoxia-inducible factor HIF alpha, as you can see over here, on the norm oxic. So when normal oxygen supplies are present, This protein is first hydroxylated, as you can see over here. Then von Hippel-Lindau factor binds to this hydroxylated HIF alpha. Ubiquitination occurs, and the protein is then, by means of this, tagged for protein degradation, and it cannot perform its tasks. However, when hypoxic conditions occur, then hydroxylation does not occur. Therefore, von Hippel-Lindau factor cannot bind to this hypoxia-inducible factor, and therefore it can now go from the cytoplasm into the nucleus, where it binds to hypoxia-responsive elements, and this increases the production of different kinds of proteins, one of them being carbonic anhydrase 9. I should note that carbonic anhydrase 12 are most likely expressed by other means, and estrogen receptors may play a role. So what I see, what I described here, are holds true for carbonic anhydrase 9. Okay, so the structures of carbonic anhydrase 9 and 12 are shown here. This is a schematic overview on the left and right. This is more or less three-dimensional like overview. This is the cell membrane. So if you look at 9, you see that at the N-terminal domain, there is this proteoglycan moiety, which you also can see over here. So there's basically a sugar coating on the enzyme on the extracellular side. Then you have the catalytic domain, which you can see over here, the transmembrane domain, and the C-terminal domain, which is cytosolic. So this is the extracellular side, the intracellular side, and this enzyme the binding pockets are separated, are located at the middle of the monomer. So the binding pockets are not at the interface of the dimer, as you can see in beta-type carbonic anhydrases. However, it's a dimer, there's a cysteine bond, and so per enzyme you have 2 catalytic sites present. And this holds true for carbonic anhydrase 12, the huge difference being that this sugar coating is not present, or to a lesser degree, on carbonic anhydrase 12. So in the 1990s at Istanbul University, Professor Aydın Salman and his crew synthesized this specific compound, which they found showed some antifungal activity. And later on, in 2008, Özlem Güzel and Claudio Supuran worked together and found that this compound also showed potent but unselective carbonic anhydrase inhibition. So it has low nanomolar inhibition for carbonic anhydrase 1 and 2, the off targets that we don't want to target, but also 9 and 12 are targets that we want to address with these carbonic anhydrase inhibitors. And they also obtained a crystal structure showing how the compound binds to the active site, and you see the interaction with the active site zinc ion and what kind of ligand protein interactions occur. And the PDB structure was using their work. And so this was basically our inspiration to try to obtain selective inhibitors against 9 and or 12, which were not or less effective against 1 and 2, preferably. Also the rest, but we didn't have the means to look at them. But also we checked whether we preferred their activity against the widely expressed 1 and 2 was much lower. So I am sorry. Okay. So what we then did was the following. Our question was this: can we generate selective carbonic anhydrase inhibitors using these using in silico combinatorial chemistry procedures? So basically, we have the crystal structure at Istanbul University, also at University of Florence. There are very good pharmaceutical chemists that had ideas on how to improve selectivity. On our side, with molecular modeling studies, we did some dockings, preliminary studies that gave us indications on how to optimize the molecules to gain subtype selectivity. I can't discuss that in detail now, unfortunately, because we hope to be able to patent these kinds of compounds. But what we did is these three regions, which are basically the entire molecule, of course, were changed according to our in silico studies, and also the sulfonamiters was allowed to change its position. And using these in silico combinatorial chemical approaches, we designed, or we generated, I should say, a targeted library of potential carbonic anhydrase inhibitors, or I should say potential selective carbonic anhydrase inhibitors containing more than 100,000 molecules. And this targeted library was then fed into our in silico screening procedures to try to obtain selective ligands. So the next question was, we have generated this targeted library. Can we identify compounds that targets the active sites of 9 and or 12? And this is how it works. So this is the proprietary targeted library of more than 100,000 compounds. So the first step was, can we have performed an ensemble docking with stepwise rescoring. So ensemble docking, ensemble means basically a collection. So what we did here is we had a collection of carbonic anhydrase 9, preferably with co-crystallized high affinity ligands. So what we did was we selected different structures with high resolution, no mutations in the active site, no missing residues in the active site. So the active site should be completely resolved. A competitor, or I should say an inhibitor with preferably high inhibition. These kinds of structures were then compared to each other. We superposed them, and only if we saw, significant differences in the binding modes or in the rotametric states of the amino acids composing the active site, we selected them for this ensemble docking. So by doing this, we had approximately, if I remember correctly, 9 different carbonic anhydrase 9 crystal structures that we used inside the docking. So the first docking step was with the HTVS scoring function. This is the Schrödinger package, by the way, with the Maestro using Glide. We performed, of course, retrospective dockings to see whether the post-generation was okay. So the first step was to dock with HTVS, which is very fast, and we showed that it was able to predict the correct binding modes of the co-crystallized ligands of the used crystal structures. And this HTVS, which is very fast again, was used to dock into all the selected carbonic anhydrase 9 crystal structures. And then the poses that we generated, the top ranked 10% of the poses that we have ranked with HTVS were only rescored. So we kept the poses, we only calculated new scores with SP, which is slower compared to HTVS, approximately 5 times. And then again, top scoring poses were then used to be rescored with XP, which is again much slower, but somewhat more accurate compared to SP. So we narrowed down the compounds. And then in the final step, we did a rescoring with MMGBSA, which is much slower compared to XP and also the others. And during this rescoring, we also allowed the amino acids of the binding pocket to relax. So by using this step, we basically implemented a filter that was allowed to filter out all compounds with docked poses that were not expected to be stable during energy minimization. That's basically what we have done here. And by doing this, we then narrowed the library down to pre-hits. I am calling them pre-hits, compounds that are expected to bind to carbonic anhydrase 9. And the same procedure with a different number of crystal structures was performed for carbonic anhydrase 12. And again, it led to pre-hits for carbonic anhydrase 12. So then the next step was, okay, now we have performed the screening, we have defined, or we have selected the compounds that we expect to be active to bind to either 9 and or 12. Then the next question was, of course, well, would these compounds also bind to 1 and 2? And if yes, we would like to filter them out, of course. So we take the pre-hits of 9 and 12, and the pre-hits were then docked inside 1 and 2 using exactly the same procedure as I described in my previous slides. And then we know the off-target hits. So which compounds are expected to bind to 1, which compounds are expected to bind to 2. And then this simple formula occurs. This is our selection criteria. So basically our hits, so the compounds that we select for molecular dynamic simulations should be the pre-hits that are expected to bind to either 9 and or 12, and minus the off-target hits. So the compounds that are also expected to bind to either 1 or 2 are then subtracted from this, and this gives our hits. So basically the hit collection are the compounds that are expected to bind to 9 and or 12, and at the same time not to 1 and or 2. Yeah, so this is our definition of hits, and this is the procedure that we have performed, or I should say my PhD students. So then, of course, some ligand selection criteria for MD. In the previous slides, we discussed that we looked at scoring, but of course, especially at the first step, I'm sorry, I'm going the wrong direction. At the first step, when I do the docking with ATVS, and here where I minimize the pose, I of course need some selection criteria to define or to check whether I want to use these compounds in my further investigations with MD simulations, 250 nanosecond per complex. So I want a stable pose of the MMGBSA and also preferably negative, as much as negative as it can be, MMGBS value for the score. But this is of course not enough. You also need some either visual inspection or some kind of fingerprint analysis, some automatic analysis. And that was performed, a combination of both basically, where I investigated the complementarity in shape, hydrogen bonding, electrostatics, and hydrophobicity. I am talking about complementarity. So we are not only looking whether these two are compatible, but we are also looking at possible mismatches. For example, hydrogen bonding. Before binding of the ligands to the binding pocket, they are in a watery environment. They have interactions with water molecules, of course. The same holds true for the active site. When these two bind, the hydrogen bonds with the water molecules need to be broken. And then you bring them together. And then the idea is that the hydrogen bond should be preferably restored again. But instead of the water, there's either a ligand or a protein to the original ligand and protein. So mismatches are that you have a hydrogen bond donor, for example, in your ligand that faces a hydrophobic region where you don't have a hydrogen bonding partner. So these kinds of poses tend to occur in docking procedures, and these are not very what you prefer. So therefore, we also check for mismatches between ligand and protein. And using this, we select the compounds for MD simulations, 250 nanoseconds. After the MD simulations, we performed, of course, RMSD calculations on the active site or the protein in general, the ligand. This shows us whether the pose stays stable or not, or whether it dissociates from the active site. And if we find that the ligand stays in the active site, then we are, of course, interested in ligand-protein interactions during the entire 250 nanosecond simulations. So this gives us both an ensemble to perform new MMGBSA calculations upon. It gives us different kinds of microstates, of course, and also we can see how frequent the ligand-protein interactions are, and this gives us a clue for whether the dock post is stable, and we can really expect the inhibitor to be active or binding potently to the target enzymes. So stable poses, once obtained after the 250 MD simulations, were subjected to GBSA calculations. This take 48 hours per simulation, so it takes a long time, especially if you have a lot of simulation that you had to do. Therefore, we need some selection criteria, which is here. After the MMGBSA calculations, we have a general idea of the calculated binding energy between ligand and protein. The value on itself doesn't say much, of course, because there's also a high degree of error inside these kinds of prediction tools. But what we do is we compare it to the results of co-crystallized ligands. So we took the co-crystallized ligand, so the crystal structure with the co-crystallized ligand that were used in the ensemble docking. We performed 250 NS-MD simulations, and we performed all the analysis, as you can see over here, including the MMGBSA energy calculations, and then we compared, and this gave us an idea whether or not the compound that we have identified here was expected to bind selectively to 9 or 12. So I'm now showing some of the results. These are the retrospective dockings. These are necessary before you start your docking procedures, because you need to be sure that your docking procedure is able to correctly predict the binding pose of a ligand. And for this, what we do is retrospective dockings. So we take the crystal structure, we take the co-crystallized ligand out, we dock it back, and then compare whether the docked post and the co-crystallized post of the reference ligand is similar. And as you can see over here, in two different kinds of green, you see that the ligand-protein interactions are predicted correctly. For this, even though there are some minor differences, the ligand-protein interactions are predicted correctly. And this has been performed for all crystal structures that are used in all ensemble docking. So for 1, 2, 9, and 12. So that was more or less 50 crystal structures that we had to subject to retrospective docking validation studies. So now I am discussing just shortly an MD simulation of a hit, that I will call hit. I'm not showing any two-dimensional ligand structures. So unfortunately, I'm just showing you these results. This is in blue on the left side. You read the RMSD value of the alpha carbon atoms of the protein, and you see that they are around 1.2, 1.4 angstrom. So the protein conformation stays stable during the 250 nanosecond simulation. In red, which you can see read on the right-hand side, you see the RMSD of the ligand-heavy atoms. And you see that during the first approximately 50 nanoseconds, we have a 1.2 angstrom RMSD value. It increases a little bit, and the remaining 150 nanoseconds stays between 2 and 2.4. So this means there has been a rearrangement, a small rearrangement of the ligand inside the binding pocket, and that you also see here back in the MMGBSA energy calculations. So you see that the energy goes a little bit down. So the binding is a little bit less what you hoped for. But still, it overall, when also compared with the reference ligand, we had an idea that this was acceptable, and therefore we selected this for synthesis. And here you see all the interactions of the specific ligand with the amino acids of the active site. I will not discuss this too much in detail, but it gives us the following results. Several compounds have been synthesized. In red, I have indicated the Ki values in nanomolar concentrations, the Ki values that were below 100 nanomolars. This is for one structure, scaffold of compounds, arbitrary numbering, and you see that several compounds, for example, these are not what we hope for, because even though they have very good, strong inhibition of the target 1 and 12 enzymes. They also show reasonably strong inhibition of 1 and 2 or just 2. So this is not what we aimed for. And then an example of a compound that shows moderate selectivity for 9 compared to the rest. It's a moderate one, not very strong in this case. Similar story for this compound, which shows somewhat more stronger but still moderate inhibition of 12 compared to the rest. However, if you continue, we see much nicer molecules. For example, this one shows strong selectivity for 2. These compounds also, 12, I should say, I'm sorry. These compounds also strong selectivity for 12. These compounds also. These two compounds, strong selective, and these compounds, strong selectivity for 9 and 12 over 1 and 2. And here another scaffold, the same holds true here again. We find some interesting compounds that show moderate to high selectivity towards 9 and or 12. So that brings me to the conclusions of the study. So using these ensemble screening with successive rescoring procedures and also in silico combinatorial chemistry, ligand generation procedures, I could share that we have constructed a targeted library of potential CAIs containing more than 100,000 molecules. These are unique molecules. These are not tautomers or isomers from each other. Unique molecules, we performed some docking studies and we selected 57 molecules belonging to 3 different scaffolds for synthesis. These compounds were synthesized and then tested, and this is the outcome. 4 compounds show intermediate selectivity towards 9. 11 compounds show intermediate to strong selectivity for 12. 25 compounds show intermediate to strong selectivity to 9 and 12. And that was the outcome of our virtual screening procedure. And finally, of course, our virtual screening was not a miracle. There were many things, many compounds we expected to be active but were less active, or we expected to be selective but they did not show any selectivity. So there are still, even though the outcome, I'm happy with the outcome, there's still room for development and optimization. And for this, in our future work, we want to predict water molecules that may be important in ligand-protein hydrogen bonding interaction. So we would like to predict water molecules that basically act as a residue inside the active site and which mediate ligand-protein interactions. And physiology shows that these kinds of water molecules are very important. And also entropy changes are very important in the binding affinity. And I'm talking about entropy changes of ligand, protein, and water molecules upon ligand binding. And there are some expansive computational tools to do this. And in our future work, we definitely want to include this in our virtual screenings. So then my final slide, I want to acknowledge different very valuable people, the PhD students that that performed most of the work, Muhammed Travelli and Kübra Demiryazıcı from Istanbul University Faculty of Pharmacy, Özlem, their supervisor. I am the second supervisor of these students. She was responsible for guiding them with the pharmaceutical synthesis, because they not only performed the molecular modeling, but also the synthesis of the compounds. And Claudio, I thank him for his valuable support, because he did all the enzyme inhibition assays as I presented over here. And finally, I would like to thank TÜBİTAK for providing the research grant under this number that made this work possible. So thank you very much for listening. Simone, I give you the floor back.
Thank you so much for your presentation, and also for this exhaustive analysis and overview of the world of the carbonic anhydrases. We are out of time, so there's no room for question for you, but I'm very delighted of your presence today, also because we are friends and collaborators about this field. So I hope that you could also contribute to the journal with one of your articles related to this topic, which is very, very interesting and suitable for anti-cancer agents in medicinal chemistry. Thank you so much for being here with us. And I will leave the word to my colleague, Ivana, in order to present another speaker for today. Thank you.
Okay. Next speaker is Professor Mangalaju. Good morning.
Hello, everybody. It's a pleasure for me to be here with you. And I wish to thanks to Ivana and Simone for their kindly invitation. And today I decided to present you some of our stuff that we are doing here in Iași. You have here a beautiful view of my university during the night time. So if you are planning to pay a visit in Iași, try to come here. It's a very nice place, and we are trying to do a good science, at least an honest one. So we are working in nitrogen heterocycle area, and this is not only because of the beauty of the chemistry involved, but also because of their applications. And today, according with the theme of this meeting, I will try to present you some results in the area of anti-cancer compounds with aza-heterocycle skeleton. We are as design, we are using to approach the molecular hybridization approach and the fusion of different pharmacophoric parts via chimeric approach. As far as the anti-cancer compounds, we are interested to target especially the DNA helix. And here we have some results in compounds which are doing covalent bonds with the DNA, and compounds that are targeting DNA via electron transfer. But mostly our work is related to designing and synthesizing compounds that are interacting via non-covalent bonding interaction with DNA, and also via a mixed mechanism, compounds that have in the same molecule different pharmacophores that are using to interact via bonding and not bonding interactions in the same time. Also, we have some tubulin inhibitors and topo 2 compounds, and I will give you an image about all of these compounds. So concerning non-covalent bonding interaction, we are focusing in obtaining typical compounds, alpha helix mimetics, and J quadruplex. Okay, let's start with the first class of compounds. So we decide to synthesize some hybrid compounds having in the same molecule of 6-member ring aza-heterocycle and a 5-member ring aza-heterocycles, so a p-deficient and a p-rich system connected via different linker, especially with amidic linker, and with different length of the alkylic part from this side connected with the NH part of the molecule. And using this strategy, we obtained different classes of compounds with pyridinic and with quinolinic, isoquinolinic system, combining with imidazolic, benzimidazolic parts with different length here in the alkynic part. And then from this purine compounds, you're obtaining the salts, and then using our experience in ylide chemistry, nitrogen ylidic cycloammonium chemistry, actually, we done different cycloaddition reactions, and we obtained these new classes of compounds having in the same molecule this fuse it fuse it imidazole or benzimidazolic system. How we proceed, we try to use a simple chemistry. For instance, in the case of this pyridinic system, we do a simple acylation, and then using this activated halogen from here, we do an alkylation to imidazole, and then obtain the base compound in the case of both imidazole and benzimidazole. Then we do the alkylation to the second nitrogen. And in the case of imidazolic system, we obtained the salt, which we was trying to do the cycloaddition, but we was not able to obtain anything. Our opinion is that this, in the conditions that we are using in the reactions, perhaps this imidazolic system decompose. In the case of benzimidazolic system, the ylidic system react with compounds with double and triple bonds, and these compounds could be obtained. That was our first attempt. And today I decided to focus more to these two types of compounds. This one that have in the same molecule a quinolinic part and an imidazolic or benzimidazolic part linked via this amidic linker, as I already told you. And also in equal measure, we was interested about the influence of these two types of radical which are anchored on this phenylic ring. Also, we have some research in the area of compounds without this phenylic part, with OR or simply with air. And the second are compounds derived from these salts, and in which you obtain this pyrroloimidazolic structure here. How we proceed? Using the strategy that I told you, from 8-aminoquinoline, we did the acylation. For instance, with these compounds having in the alkenic part N1 and N2, but we have also with different CH2 until on 6 CH2, so N equals 6 here. And we get the first class of compound, then we do the alkylation, and we obtain the first class of compounds. We are trying to do cycloaddition, and again, the reaction doesn't work. So the same rule was applied that in the previous case, perhaps it decomposed these compounds. But in the case of benzimidazolic system, it was possible to do the cycloaddition reaction, and we get the desired compounds, this one having the benzimidazole in the molecule. But also, as you will see, we get some other compounds which in a way make us not happy at the beginning, but finally we was happy like that. So how the reactions are working? Initially, we was trying to transform these salts using this ylid chemistry here to do cycloaddition to this one free dipole with different dipolarophile, and I choose here the MAD, using free ethylamine and as basis in order to realize the deprotonation from here to take the hydrobromic acid from this molecule. And when we use triethylamine in we was trying to use also conventional conditions, that means thermal heating, and also we did this reaction under ultrasound irradiation. So what it was happening? When we use triethylamine under conventional thermal heating, the reaction, it doesn't work. And we was quite intriguing because normally it should work, but it doesn't work simply. And we have some explanation. I will not get into detail from here. But when you use the ultrasound irradiation, we was able to obtain the desired compound, which is this one, the pyrroloimidazolic system. But together with it, we get also two other compounds. This one from the left side, which is a dihydropyrrolobenzimidazolic system. And actually, this is the intermediary in the reaction. So when the first step of reaction is the cycloaddition of this triple bond to here, and it obtained this one, and it seems that it is enough stable to remain in the reaction. Normally, this one should occur and should go in this fully aromatized compounds via an oxidative process, but in this case, it doesn't happen like that. And we obtaining a strange compound, this pyrazilic system, to which I will come back. When we saw it first time, it was quite difficult to establish the structure. But finally, we managed to do it using including X-ray diffractions, and we are very sure that this is the compound. And as you will see, we have an explanation for the obtaining of this. But the selectivity of this reaction is not good because we have a mixture. So we was trying to improve the selectivity of reaction. So we changed the catalyst and the solvent. We use this butylene oxide both as catalyst and solvent in the reaction. And what was happening? Under ultrasound or conventional thermal heating, we obtaining only two compounds, but again, this partial aromatized compound. So we was not happy also, the selectivity was not good also. So this is why we use catalytic reaction, use this TPCD as catalyst, which realizing the oxidation of this pyrrolo system, and in this case we obtain only the aromatized system. So we was happy at the end, but it was, believe me, quite a lot of work in order to do this synthesis. Then we send the compounds for anti-cancer test, and we send it to National Cancer Institute in USA, that and you know very well that they are performing the this assay on a panel of 60 type of cancer cells, and here you have the some of the results that we obtained. Actually, as you could see here from this slide, there are these compounds that are bearing in this para position of phenyl ring, another phenyl ring here. And it seems that these are the most active compounds that have in the para position a phenyl ring. And of course, it also depends, as you could see here, the structure of the initial compounds. That means if the compounds have a imidazole or a benzimidazole system, and the number of the CH2 group from the molecule. The most active compound is this one that have a benzimidazole, a phenyl group here, only one phenyl in the para position, and also only one CH2 group here in the amidic linker from here. And as you could see, that the compounds are quite very active against leukemia, 3 types, and the cytotoxic and the cytostatic effect is 100%, and the cytotoxic effect is 35%. And as you could see here, the same very good results to the other 2 leukemia, to this non-small lung cancer, and also to colon cancer, to CNS, central nervous system cancer, to melanoma, to ovarian cancer, to breast cancer, good results. Oh, sorry. So it seems that these compounds have a quasi non-selective activity, and this is why actually this compound passed in the second stage of the test, the so-called 5-dose assay. That one, it was the 1-dose assay, you know very well. And as you could see from this table, to some particularly type of cancer cells, the compounds are, this compound is very active, is in the area of 0.1 or 0.2 micromolar, so very closely to nanomolar, which means that it's a promising leading candidate for obtaining other compounds with good anti-cancer activity. We was wondering about the mechanism of this compounds, and of course we have had from previously work some idea, and we done some molecular docking, not on the same level of our previous speaker done it before, but to a certain level, and we took into consideration three types of interaction. Interaction with DNA chain, the J quadruplex interaction, and the interaction with a complex made by deoxygenase enzyme and DNA string. And here are the results. I will not get into detail. It was a full molecular docking. Here are the interaction with the different type of amino acids. Here are on the upper part are the binding sites in which the compound are going in these three type of models. Here are the interaction with the first receptor, the interaction with the second receptor, the J quadruplex, and the interaction with the third receptor, that one which is the mixture between deoxygenase and DNA strings. As you could see here, the interaction are between the molecule and the DNA strings are in both sides. And here are exactly the interaction with the amino acids from enzyme. So to summarize, having in view the results that we obtaining, so that means the total complex energy and the dissociation constant, the constant affinity. So we decide, and the results confirm it, that the most interaction will be with the receptor free, this one. So the mixture between the deoxygenase and DNA string. So perhaps our compounds interact with both. Then we move to another type of molecules using the chimeric approach, in which we was trying to combine the potential of phenanthrolene, different type of phenanthrolene, 110, 47, 46, with parahalogeno benzoyl moiety, because in previously paper work, we noticed that having a halogen here, especially a chloride, and in some extent a bromide also, this allow to our compounds to interact with some putative binding sites, and the activity of compounds increase very much. And we obtain a compound as this one, the chimeric one, in which you have pyrrolo phenanthrolenic systems. And here are some results that we obtained some years ago. From here we start. We done the same lead chemistry with this triple bond compounds, for instance, and we obtaining this sort of compounds, fully aromatized or partial aromatized. We obtained nice crystal at the end, and here it is depicted, the X-ray structure, of course. Here are some reaction made with alkenic structure. As you could see, typical one or cyclic one. And again, we obtain this sort of compounds, de-aromatized, this saturated or partial saturated, or here again, fully saturated with 2, 4, 5 rings in the same molecule and the X-ray structures. And having in view that results, we decide to move a little bit further and to combine the potential of the phenanthrolenic ring and to merge it with another heterocycle, a pyridinic one, or as a quinolinic one, or a thiazolic one, via this amidic bound, the linker. And also, in some cases, we obtain it this phenanthrolenic ring with triple bond, which allowed us to do some click chemistry, and we did it. And after we get this sort of compounds with different substituents here, and I will show you very soon, we decide to transform it in salts, and we obtain it in some cases monosalts, and in the most cases bis salts. According, it was a question of heterocycle that you have it here, if you was able to obtain bis salts and monosalts. Why we did this? Because you can imagine that to these big molecules was we have had some problem, we encountered some problem of solubility, and we are trying to increase the solubility of compounds. And also, you know very well that in some cases the action of an ion, counter anion, could contribute to the increase of activity. And for the compounds with triple bond, we did click chemistry when we obtained this phenanthrolinic with triazolic system in the same molecules. And here, linked it to the triazole, we have it again, this para R benzoylic systems. So here are the results concerning the anti-cancer activity. And as you could see here, we have here some, we was trying to obtain here typical J quadruplex compounds for in order to realize the penetration of DNA to in the J quadruplex area in the telomeric system. And here are the results. Here are the compounds which are not transformed in salts, the compounds that are transformed in bis salts and in monosalts. And these are the compounds with the triazolic system. These are of type AC. What we noticed from the results that we received from National Cancer Institute, that this compound with the pyridinic system substituted in the beta position with this moiety have a quasi non-selective activity. And also the bis salts have also a quasi non-selective activity against different types of cancer cells, as you could see here. So it seems that it counts the nature of heterocycle, and also it's count if you have a bis salt or a monosalt. The monosalts prove to be not so active, and also the compounds with triazolic system not active. We was trying to see which will be the most potent interactions, if it will be in the J quadruplex area, and we took into consideration also interaction with the minor growth. And as you could see here from the energy binding and dissociation constants, both are possible, but with a plus on the J quadruplex interactions, because the binding energy are smaller and the dissociation constants are higher. Okay. We have some good results, I could say, in the area of compounds that are acting as fenstatin analogus. And in this respect, we was focused, of course, in this pyrrolo pyridinic or diazinic systems, because we was trying to modify this part of fenstatin, and to using the isosteric approach, the non-classical isosteric approach, we decided to remove, actually, yes, to remove this phenylic part with a non-classical isoster, this pyrrolo azinic or diazinic system, keeping this part, this trimethoxy part, more or less the same. In some cases the same, in some cases with only with two substituents in different positions, 14, 14, or 15 part. How we proceed? We use, for instance, the pyridinic or the bipyridinic system. We done the alkylation, and then we done the cycloaddition, and we get this pyrrolo bipyridinic systems. In the case of pyridazine, we obtain some pyrrolo pyridazinic systems. And here are the results. We done some more other reactions, but I will not get in all the reactions because I'll be out of time for sure. And here are the results. As you could see here, in the case of some compounds, particularly these compounds with pyrrolo-bipyridinic system, the activity of compounds are quite good according with the substituents that we have it in, but also even the compounds having the diazinic system, this 15B, have also a nice activity in some cases, as you could see here, 100% inhibition of cancer cells. And here are the results for the 5 doses. And as you could see here, in the case of bipyridinic system, they are very active to 0.04, 0.05. So this means that they are almost in the nanomolar. Here it is 48 nanomolar concentration. So this means a very good activity against non-cancer, colon cancer, CNS cancer, and melanoma. What we also notice, and we think that it's also a very important clue, that the pyrrolopyridazinic compounds have a selective activity only against melanoma, as you could see here, to 0.30 nanomolar. So I think that these 2 groups of compounds are also interested to be further studied. And because of this, we decide to improve and to enlarge the area of the compounds that we obtained, and we have had in mind this 1, 2, 3, 4, 5 type of compounds in order to be obtained. And how we did it? Again, we use our experience in dilute chemistry. For instance, in the case of pyrazine, we get this pyrrolopyrazinic system. In the case of pyrimidine, substituted with this parachlorophenyl, we get this pyrrolopyrimidinic systems. In the case of quinoline, we get the pyrroloquinolinic system. In the case of benzo-J-quinoline, we get this pyrrolo-benzo-J-quinolinic system. And in the case of isoquinoline, this pyrrolo-isoquinolinic systems. Okay, so let's see which are the results. So if you are looking here in the table, and here I just pick up some of the data that the guys from National Cancer Institute sent it to us. You may notice that compound with pyrroloquinolinic structures of this type and pyrrolo-isoquinolinic structures proves to be the most active. As you could see here, they're active 100% growth inhibitions for this compound and this compound to different type of cancer cells. Look here. So quite a non-selective activity. And because of this, they pass in the second stage. And for instance, in this pyrroloquinolinic system, you could see that to some particularly cancer cells, the activity is also in the area of nanomolar. You see 50 nanomolar, 35 nanomolar, 25 nanomolar, which is a very good asset for this sort of compounds. We was trying to see, we done some molecular docking, trying to see which are the most probable interactions with the receptors. And we took into consideration, of course, the interaction with the colchicine binding site, according with the literature data. And as you could see here, we know exactly at this time how these compounds are binding in the different parts of the colchicinic site of compounds. It is depicted here. I will not get into detail. And here it is my team. As you could see here, I have smart and bright guys and girls in the team. I have to send also to Romanian Ministry of Education for funds, and also to European Union for some complementary funds. And thank you very much for your attention.
Okay, thank you. Good morning, everyone. And I'm very honored to be here as a speaker. And thanks for the invitation to the committee. Today, I'm gonna present what is my little experience in the field of inflammation and what I think is the fascinating world of inflammation, and how I am working hard to try to give a contribution to this field. So first, what is inflammation? Inflammation is very generally speaking is our body's natural response to protect itself, and from a molecular point of view, these are the unlock my pointer. Okay, these are the characters, the main players in the inflammation downstream. They are monocyte macrophages, which are cells that increase upon acute inflammation. And officially, this is a natural immunology definition. Inflammation is a response to stimulation by invading pathogens or endogenous signals such as damaged cells or cancer cells also, that results in tissue repair or sometimes pathology when the response goes unchecked. So when everything goes checked, what happens? Our cells surround the area to protect the area, and the injury stops. So when it goes unchecked, this is our crossroads. Injuries could go deeper, and infections could be deadly, or the inflammation can go chronic, and a low-grade chronic inflammation can occur. So when everything goes checked, after a bound, after an infection, after a viral or a bacterial, a fungal infection, it could be a chemical irritant, it could be ischemic stroke, traumatic injury, or even a frostbite or a burn or allergy. Our monocyte, our circulating monocytes activate, and there is an extravasation, and they go to the damaged tissue to cure this acute inflammation, and everything goes checked. When everything goes unchecked, what happens? When everything goes unchecked, what happens? It happens that, as I said, chronic or low-grade inflammation occur, and from the expert of the field, this chronic low-grade inflammation, it's called the silent killer, because it could be it could occur cardiovascular disease or metabolic disorders, autoimmune diseases, neurodegeneration, even mental disorders or neuromuscular disorders, and cancer. If you see this scheme, I put a one-side arrow in the acute inflammation, because, as I said, if everything goes checked, our body can fight the inflammation, and wound healing occurs, pro-resolving infection occurs, and everything is eliminated from our body. If this inflammation goes unchecked, all these chronic disorders can occur, and of course, also there is a double-headed arrow, because this is a burden. This is something that can occur in parallel. So cardiovascular disease in parallel with metabolic disorders, autoimmune disease, cancers, neuroinflammation with mental void. It is to avoid this low grade, is to avoid that our immune system start to be dysregulated. what are the currently treatments for low grade or chronic inflammation? Currently, steroidal and non-steroidal anti-inflammatory drugs, analgesics, or other pain medications are the most commonly used canonical treatments or classical treatments for inflammation in a clinical practice. However, long-term use of these drugs, as it is largely known, can cause kinds of adverse reactions at skin level or liver level or gastrointestinal level. So while rational use of this drug is allowed, of course, compounds with highest safety and as much as possible low toxicity should be used. And over the last decades, natural compounds have attracted strong interest in this sense, not only because they have a wide range of pharmacological applications, but also for the possibility to chemical modifications to improve pharmacodynamic and pharmacokinetics. And natural products have a wide range of applications and activity and potentially low toxicity. And of course, some severe symptoms are difficult to be treated with natural products, but they are much more effective for relieving pain in the early stages of inflammation. In recent years, great progress has been made in the development and resolution of mechanisms of chronic inflammation and in diseases related to this chronic inflammation, and the use of these products to alleviate pain symptoms, of course. Research is nowadays committed to highlight molecular mechanisms underlying the use of natural products and to find a potential molecular target, because in a lot of these pathologies, such as, I don't know, in the next slide, I will talk about tendinopathies, for example, there is no an exact and a unique molecular target. So current studies have found that natural products endowed with anti-inflammatory activities include flavonoids, polysaccharides, polyphenols, alkaloids, terpenes, natural pigments, plant volatile oils, and similar. There is a plethora of literature reporting on their anti-inflammatory activity and the anti-inflammatory value and potential molecular mechanism of action. Studied is the NRF2 KEAP1 hemioxygenase 1 molecular axis, and fortunately is what I knew from 10 years during my research experience here in Chieti and above all in Regensburg in cooperation with Professor Helmut Schweikel from the University of Regensburg. Here it is, NRF2 is my idea of the good and bad of inflammation. As I said, I've been studied it from 10 years and exactly, yes, 10 years. And it is still, but let's make the proper introductions now from a molecular point of view. Cells have developed multidimensional adaptive mechanisms to regenerate a balanced redox homeostasis, and NRF2 represents the master regulator of cell responses towards oxidative stress and oxidative stress-related inflammation in immunocompetent cells. It is a helix loop helix basic leucine zipper transcription factor, and it is inhibited in the cytosol because of the binding to its natural physiological inhibitor, which is KEAP1. It is inhibited also due to a constant constant ubiquitination and proteasomal degradation. As you can see, this is the inhibitor KEAP1 here. NRF2 can be released from KEAP1 by oxidative stress, and so escapes degradation. So here it is, KEAP1, and it links to the ARE. NRF2 can be unbonded from KEAP1 by oxidative stress, so by, in this case, ROS. And it is unbounded from KEAP1, and it links on the ARE on the promoter region. And the main function of NRF2 is to activate downstream target genes coding for enzymatic antioxidants, such as which metabolize directly IRS. So as an example, there is SOD1 catalase or glutathione peroxidase. And their function is assisted by hemioxygenase 1, which is auxiliary to NRF2 function, and they cooperate to regenerate the intracellular pool of GSH, so the glutathione. So this is what we try to demonstrate in this paper with Professor Schweikel, and we use a composite material, which is HEMA, 2-hydroxyl metacrylate. And this is a common composite material which is used in restorative dentistry. So we study this phenomenon on cells of the oral cavity, in particular on macrophages. In this other paper, we try to demonstrate this pathway during infections. As you know, the LPS or bacterial product, they are a very strong pump, so pathogen activating pattern, and they link to CD14 and toll-like receptors on the macrophage membrane and activate the NF-κB downstreams, which activate in turn the iNOS and the production and the generation of peroxynitrate from nitric oxide and superoxide anions produced from NAPH NOx enzymes. So what we observed, this is a very full diapo, but what we observe is that the oxidative stress in the presence of metacrylates produces diverse type of ROS. It depends on the time of exposure, and it depends on the type of stimulus. What we found is that in the very first moment of the exposure of an irritant, the increase of ROS is mainly hydrogen peroxide, and this increase allows for the oxidation of sulfhydrylic groups on cysteines, thus modifying cell signaling via protein oxidation, or formation of hydroxyl groups in the DNA, and consequently lead to apoptosis. Cells, in this case, immediately activate, and when I say immediately, I meant within an hour, the expression of NRF2 and its translocation to the nucleus, augmenting cell viability. In our second paper was born from a preliminary observation, which is that when we exposed LPS, when we exposed macrophages to LPS and HEMA in parallel, we had a strong decrease of the release of cytokines, pro-inflammatory cytokines, above all TNF-alpha. And this is very weird because the habit is that when we expose to LPS and then we expose to an irritant, there must be an increase in cytokine release. So in a clinical practice, it is very dangerous that a patient that have an infections from gram-positive or gram-negative undergo a surgery for restorative dentistry, and his immunity is some kind of dysregulated. So to translate it in a clinical situation, during infections of the oral cavity, it might be self-defeating to do not have a proper control of the infection. So what we demonstrated in this paper we have, I go in before, we have demonstrated that the peroxynitrate, which is enters when the LPS signaling starts, this peroxynitrate that accumulate intracellularly activate NRF2. And what is NRF2? NRF2 is, as I said, the master regulator of the antioxidant response. So the translocation of NRF2 due to the peroxynitrate and hydrogen peroxide accumulation inhibits this pathway, this NFKB pathway, and thus cytokine secretion to modulate infection and induced inflammation. So once being fascinated by the NRF2 word, I have started to study it also in other oxidative stress-related diseases in the presence of natural the infiltration of macrophages at the low-grade chronic inflammation are features very common to several inflammation-related diseases, and among others, tendinopathies are one of the most common musculoskeletal disorders, which results in disability and high social cost for how system. Over the last 20 years, there was a huge number of studies that has conservative treatments against surgery and try to underline inflammation upon tendinopathy exist. And as I said before, to date, the unique marker or markers loss of tendon healing. So we investigated, and the hospital of Pescara, this pathway, this inflammatory pathway, and using hyaluronic acid as a tool, which has been found to be efficient in alleviating pain and improving tendon function on primary tendon-derived cells in a model of inflammation. So we treat tendon-derived at low doses to increase oxidative stress. However, as hyaluronic acid, as I said, however, is effective role at a molecular level remains. In particular, this work highlighted that each hyaluronic acid preparation can trigger a variety of response. Here there is a cytotoxicity assay, the LDH release. There is a signal, there is an uptake of hyaluronic acid after, because the hyaluronic acid receptor is enhanced, the CD44. And as I was saying, there is a different response of the cells belonging to the different hyaluronic acid preparation, which differ by their molecular. It is enhanced for sure. Is here the NRF2 in the presence of hydrogen peroxide. So we found that NRF2 is upregulated also in this model of inflammation, and when we during the administration of this hyaluronic acid preparation, we have constant NRF2 expression. That NRF2 expression is present in nation and disclosing this protein as a tendinopathies. So we highlighted that tendon-derived from apoptosis by activating the NRF2 under hyaluronic acid preparation treatment. We investigated whether this could be a general mechanism underlying an anti-inflammation action in tendons. As I said before, under oxidative stress and inflammation, NRF2 directly transcribes for hemioxygenase 1, which produces anti-inflammatory molecules such as carbonic oxide and ferrous-iron and biliverdin as crucial cell responses to restore cell homeostasis. So this therapeutic delivery of carbonic monoxide, it is the interest for this administration is increasing in literature, and we, in cooperation with the University of Florence and Professor Carradori, we administered the carbonic anhydrase inhibitors, in particular active on the CA9 and CA12 isoforms, linked to a D-cobalt as a carbonyl, which is a CO releasing moiety on tendon-derived cells treated with hydrogen peroxide. What we found that in particular in the presence of acesulfame scaffolds and coumarin scaffolds, we found that there is a huge decrease of cytotoxicity in hydrogen peroxide treated cells. Here, as you can see, after 24 hours. We found that there is the cell cycle, the proliferation of these cells, we analyzed the cell cycle, is restored. We found that at the molecular level, depending on the scaffold, they have different action on at the molecular level in tendon-derived cells. So, for example, compound 2 is very active in increasing type 1 secretion from one of the major features of the tendon healing. So we must have collagen type 1 to heal tendons, and in particular we found that compound 2 and 7 enhance NRF2. Once we have understood that this molecular target can be found in other inflamed environments, not only macrophages, we came back to our first love, which are macrophages, and to test xanthons derivatives from Garcinia mangostana. The natural compounds are alpha mangosteen and gamma mangosteen, and in cooperation with the University of Porto, we have tested these xanthon derivatives, trying to demonstrate that they enhance the NRF2 target in macrophages. Here I have reported the scheme of the biological assays. So we took monocytes from peripheral blood. This is a standard cell line purchased from ATCC. We treated with PMA, which is an enhancer of NFKB and so of superoxide anions at low doses for 48 hours. And then we obtained differentiated macrophages-like cells. And then we treated with BSO, L-butyonyl sulfiximine, which is an enhancer of, we found from our previous publications that is an enhancer of NRF2. So we pre-treated cells to enhance NRF2, and then we administered LPS and to even enhance this pathway, this NRF2 pathway, and then test compounds or mangosteen at 2 doses, 1 and 10 micromolar for 24 and 48 hours. What we obtained is that there is a huge decrease in LDH released from cells. This is the untreated control, only pre-treated with BSO. So there is a huge release of LDH, so cytotoxicity, in the presence of these selected compounds from preliminary NTT. We found a decrease in cytotoxicity, and so in the LDH release, mainly with the natural compounds, alpha and gamma manganese. Is the Western blotting the soil. What we found is that NRF2 translocates into gamma mangosteen, but also in the presence. Then our colleague from the University of Porto SAR analysis 27, which is the best compound of the series, is also an electrophilic compound. So this is why it is very because of electrophilic reaction with NRF2. So NRF2 can unbound KEAP1 in cytosolids and translocate into the nucleus. What is going now? This is an unpublished material. This is what we are doing in this moment. We are testing compounds in cooperation with the University of Firenze, with Professor Fabrizio Carta and Professor Selleri. And these compounds were found to be active in inhibiting prostaglandins, the secretion of prostaglandins in previous works. So we tested these compounds on macrophages again with the same pattern of differentiation that I showed before for xanthans, and we found that the most promising compounds are compounds 5 and 8, which in particular compound 8 is able to decrease peroxynitrate accumulation in cells, and these results are comparable to a treatment with meloxicam, a COX-2 inhibitor. We found that in the presence of compound 8 and LPS, NRF2 is strongly translocated into the nucleus. So this is an index of an anti-inflammatory response occurring. What we found is that also the NRF2, what we are trying to link is the shift, the phenotypical shift of macrophages from the pro-inflammatory phenotype M1 to the anti-inflammatory phenotype M2 with NRF2. And we found that it could be true because all the markers related to the M1 phenotype, such as iNOS, for example, are decreased in the presence of compounds. And instead, markers related to the M2, so the pro-resolving phenotype, such as TGF-beta1, are enhanced and cleaved and activated. In particular, CD80, which is a marker of the M1 phenotype, is decreased in the presence of compound, and in parallel, the CD163 is increased. The 163 is a marker for the M2 macrophage phenotype. And to come back to the other diapo, we are trying also to link NRF2, the shift of the M1, M2 phenotype to a shift in the cell metabolism, because I made a little disclaimer, when they shift from the M1 to the M2 phenotype, macrophages change their metabolic pattern, and they start to produce lactate. Is a kind of Warburg environment, like in tumor cells, but in this case, this shift in the metabolism helps cells to fight inflammation, and so we can find all the markers that are very they are players in this shift metabolism, such as CD73 and PI3K and AKT. These are pathways that are involved in this metabolic shift. And nowadays we call this field of the study of inflammation metabolic inflammation or immunometabolism. So these are the players that we are studying. CD73, which is a 5-ectonucleotidase, which is involved in the shift from an ATP-driven pro-inflammatory environment to an ADP-driven pro-resolving environment. We found that it is very increased in the presence of our compounds, as well as lactate. And in this moment we are writing this paper. We are trying to link these three aspects of inflammation using these compounds as tools. Just the last one work I want to share with you. This is not proper an inflammatory environment. This is a study of material which are involved in the osteogenesis and bone healing, and these are dental pulp cells which own a stem cell population inside to regenerate dentin and bone. We found that NRF2 is activated also in this kind of models because during implantation, inflammation occurs, and we found that NRF2 can be activated also after 28 days during this experimental design, finding that NRF2 can be linked also to the decrease in inflammation in stem cells, and so it could be linked also to osteogenesis and a redox balance of differentiation in stem cells. So, concluding, we can have one motto, which is do not turn off the light on inflammation, but to modulate molecules implicated in inflammation. And we can say that the NRF2 transcription factors is a central player in the maintenance of the redox homeostasis. And we can speculate that NRF2 is a general mechanism of cells to counteract inflammation. And so NRF2 can be an innovative therapeutic target and a suitable strategy to target a dysregulated immune response and avoid low-grade chronic inflammation in different inflammation-related diseases. And thank you for your kind attention. I want to acknowledge the Professor Schweiker from the University of Regensburg and Mrs. Carola Bolai and Claudia Ba and Christine Petzel for the help in the laboratory. And this is here on the left, our group, when we are celebrating our PhD section, and I want to thank in particular the Professor Amelia Cataldi and Professor Simone Carradori, and this is the Horizon project, which allows me to do everything I show you. Thank you for your kind attention.
Thank you, Maria Lucia. Thank you for your nice presentation and a lot of data that you presented. So, next speaker, Professor Hasan Turkets. Good morning, Hasan.
Good morning, Ivana. Good morning, everyone.
Okay. So, few words about Professor Turkets. He received his undergraduate education on biology, Faculty of Science at Atatürk University. His master thesis was on drug adverse effects definition. He has two different PhDs in genetics and pharmacology from the Atatürk University and the University Gabriele D'Annunzio of Chieti-Pescara, respectively. He has a broad-based background in genetics and drug R&D. He is serving as a toxicology and epidemiology expert in the Commission of the World Health Organization between the years of 2016 and 2026. He is a molecular biology and genetic consultant of the Scientific and Technological Research Council of Turkey, TÜBİTAK. His main interests are the molecular, pharmacological, and nanomedicinal studies of Alzheimer's disease, Parkinson's disease, and brain cancers. He published more than 250 articles with more than 4,000 citations, joined more than 150 meetings, and received international and national scientific awards. Today, he presents his talk about boron-containing therapeutics, current status and future perspective. Thank you, Sam.
Thank you, Ivana. Thanks for your nice presentation of me. First of all, I would like to thank everyone who contributed the nice organization of this beautiful and then fruitful organization. A special thanks to Dr. Ivana Cacciatore for her kind invitation of me. Me and my research team have been working on boron biology for many years, exploring new and different biological roles of boron is really exciting for me and for possible further applications in biomedical area. With the scope of today's program, I will try to explain the therapeutic effect potentials of boron-containing compounds or boron derivatives with the underlying mechanisms of the results provided from in vitro and in vivo different research findings. Since I anticipate that most of our participants will be experts of in the field of chemistry or medicinal chemistry, I will try to explain the importance of these compounds for the scientists from the perspectives of molecular biology or biology perspectives, rather than informing or directing the chemical structure in the content of the presentation. Now, our presentation is composed of 5 different sub-contents, as you've seen from the slide. We will mention about first boron and human health association, and then we will evaluate the biosafety issues associated with boron exposure. Then we will use boron-containing compounds with the observed therapeutic action potential, and then we will have some brief discussion about the challenge, and then finally we will have limited time to discuss with the further perspectives. Now, the health of not only people, also plants or animals, depends on the trace mineral boron, because we consider boron as a micronutrient, which also may have key functions in the evolution of life processing on the Earth. According to current investigations, mainly they focus on the most important impacts of boron on and human health relationships. For example, their influence on bone formation, their influence on regeneration, bone regeneration, wound healing, improving wound healing process, and then the synthesis and metabolism of sex hormones, and then vitamin D, and the absorption and usage of calcium and magnesium. Additionally, boron function includes the anti-inflammatory properties that can ease arthritis pain and enhance cognitive function. It has also known that the boron have potential anti-cancer properties, that boronated compounds are being employed in the treatment of different types of cancers. Now let me give the most significant ones in the slide, but not more. Advances in the field of boron chemistry, we have expanded the application of this element in medicinal chemistry. Mainly boron-containing compounds represents a new class of or new family of medicinal chemists to use in their drug design approach. For example, you know, everyone knows the bortezomib, or the commercial name is Velca, is a dipeptide boronic acid. And then it was approved by the FDA, I suppose that 20 years ago, for the treatment of multiple myeloma, and then paved for the discovery of advanced new boron-containing drug formulations. After its approval, 2 other boron-containing compounds have been approved. Their names were tevaborol, and the other one was the name crisaborol. They are all were used to treatment of mild to moderate atopic dermatitis conditions. So a number of boron-containing compounds have been described and evaluated for a like a plethora of therapeutic applications. In my slide, you can see that I plan to describe the current status or further perspectives towards boron use in medical area. As you know, boron is essential for the growth and maintenance of the bone health, and it is really supported that it improves boron-containing compounds, improves wound healing procedure, and then beneficially impacts the body's use of estrogen, testosterone, or vitamin D. It is known that boron compounds boost magnesium absorption and mimics calcium physiology in bone. And then it is known that boron reduces the levels of inflammatory biomarkers such as high sensitivity of C-reactive protein, and then TNF-alpha, you know, tumor necrosis factor alpha. And then boron raises the levels of antioxidant enzymes such as superoxide dismutase, catalase, and then glutathione peroxidase, as well as supporting antioxidant capacity of human tissues, strengthening antioxidant defense mechanisms, and then protects against pesticide-like contaminant-induced oxidative stress and then heavy metal toxicity. Boron improved the brain's electric activity. You know, this activity is directly related to cognitive functions and then short-term memory for the elders. Now we are seeing them from the literature that boron influenced the formation of and activity of key molecules such as, for example, as adenosine methionine or nicotinamide adenine dinucleotide levels. Finally, we know that boron has preventive and therapeutic effects in a number of cancers, such as prostate, cervical, lung cancers, as well as multiple and Hodgkin lymphoma, including this main care roles due to boron and then associated with the boron health. Then boron is a vital element for living organisms. Boron and several types of its compounds different beneficial or positive effects in humans at a relatively sufficient doses. However, too little or too much intake in the required amount causes serious health problems. Its use as an food supplement to prevent boron deficiency in the body has become popular, especially in these days, it became very popular. In addition, the use of boron and its compounds in many fields, such as cosmetics, cleaning, medicine, and then electronics, war industry, and the development of new products for these fields are increasing day by day. Such reasons cause an increase in exposure, not only to individuals working in boron mines, but also in daily life. We performed a study on people who are working in the boron mine in Turkey, especially in eastern part of Anatolia, and then we noticed that the boron exposure did not induce any fertility rate among the workers. Supporting further literature evidences indicated that boron is not also toxic in a reproductive area or reproductive systems. And then besides boron and its compounds appear as candidate molecules in the treatment of various diseases. All of these factors indicate that there will be a more expansion or exposure to boron and its compounds today and in the future. For this reason, we can conclude that these compounds, which I mentioned about their possible positive effects on human health, having many benefits under normal conditions, but we will also encounter toxic effects due to increased exposure over time. As an Bertrand principles in general, we are mentioning about our courses, the Bertrand principle indicates that every material has toxic, but we have to use its concentration or the dose, applied dose amounts. So it's also possible that some boron-containing compounds can be candidates for cancer treatment through their structural effects and then cytotoxic effects on cellular mechanisms. Therefore, shows that the importance of the toxicology studies of these compounds at all times. However, developing scientific technologies will also enable these effects to be minimized. The performed animal experiments by us or any other scientist indicated that lethal dose 50 values have clearly shown that boron is as toxic as the salt we use on our tables in daily life, not more, it's sure. Boron was misconstructed because as a toxic element for animals, which retarded the growth of boron-containing drug discovery in the last century. Nevertheless, modern applications of boronic acid derivatives are attractive in biomedical applications after the declaration that boron is a probable essential element as suggested by World Health Organization. Additionally, the approval of 5 boronic acids containing drugs by the FDA has vastly impacted the use of boron in the medical area that I mentioned in our previous slide due to their positive impacts on health. So the boron use in medicinal chemistry, chemical biology, drug delivery, biomaterial exploration, pharmacological improvements, as well as nutrition, became an attractive area for use of boron in further applications. Now, over the last several decades, some boron-containing compounds found in nature or developed with chemical synthesis have been attractive as enzyme inhibitors or antineoplastic drugs and anti-infectious disease agents. In the last 5 years, boron-containing compounds have been designed to act on G protein-coupled receptors, ionic channels, membrane receptors with enzymatic domains, and we can conclude that all types of enzymes we can use this. So boron-containing compounds also are being examined as modulators of flow of genetic information within the biological systems, both on prokaryotic and then eukaryotic systems, the cell cycle, metabolism, and the inflammatory process. This modulation frequently implies more than one molecular target. Maybe this situation makes very attractive compounds of boron to be possible use for different biomedical applications for humanity. Then in this part, the presentation, in my presentation, I will try to present the biological activities of selected boron compounds whose therapeutics or therapeutic action potential has been determined so far. The number of bioactive boronic acid containing compounds reported has increased in recent years. As I mentioned, the infectious disease and the cancer are the major targets of these new therapeutic agents, and many of the already suggested compounds or produced compounds have been designed for this application. The anti-cancer activity of boronic acid-containing compounds is generally related to their ability to inhibit cell proteasome activity. This is a large and then highly sophisticated enzyme complex, as many molecular biologists know that. The proteasome is responsible for the degradation of intracellular proteins, and then ubiquitin-flag proteins for destruction, serving as a degradation of signal to the proteasome activity. So the selective and efficient degradation systems plays an important role in the regulation of which control the cell cycle and the apoptotic rates in the cells. Therefore, important therapeutic target for the treatment of cancer. The catalytic activities of the proteasome are classified three different categories. These categories are, one of them is chemotrypsin-like, second is protein-like, the other one post-glutamic peptide, hydrolase, and caspase-like. Yes. These catalytic subunits are open to the targets or most boronic acid-containing compounds developed against of some cancers. Besides proteasome inhibitions, boronic acid-containing compounds were also described as anti-cancer agents that acting on different cancer types. For instance, fulvestrant derivative containing boron was reported as a promising target for the clinical investigations because it is super oral bioavailability values. in mice compared to the parent drug, fulvestrant. Specifically, boronic acid containing compounds are selective estrogen receptor down regulators. So they bind to estrogen receptor competitively and down regulate it in both tamoxifen sensitive or tamoxifen resident cancer cells in breast cancer cases. This data is really interesting that the boronic acids will be provide a good inspiration for the novel treatment of breast cancer strategies. Boronic acid compounds have also been widely used in the design of, interestingly, beta-lactam or beta-lactamase inhibitors. The use of boronic acid containing compounds as beta-lactamase inhibitors has a huge advantage over commercially available beta-lactamase inhibitors, very well known, like as clavulanate or tazobactam. Really, these boronic acid containing compounds have been shown to have more active than these commercially available ones. Similar to boronic acid compounds, different kind of this family, boron compounds family, for bacterial infections have been designed as an antiviral agents. For example, GSK, as I recorded last week, GSK introduced a novel boronic acid containing compound as an inhibitor of the hepatitis C virus. So RNA-dependent RNA polymerase interaction. This enzyme is responsible for synthesizing new viral RNA able to inhibit hepatitis C virus replication. The most promising, maybe the most promising results in this compound family came from the in vivo study, because influenza A virus infected mice treated with the boronic acid containing compounds. I remember the dose 10 milligram per kg showed better survival rates than mice treated with the standard drug oseltamivir. As I repeat, I think it's okay. Oseltamivir. Yeah. Therefore, boronic acids recorded as an promising against infection disease, cancers, and as an antiviral agent until now. The second main groups of boron is benzoxyborols. The second family, whose therapeutic potential has been evaluated after careful review of the relevant literature, emerged as benzoxyborols, in my opinion. As I learned from my chemistry team, the boron carbon bond in the oxaboron ring has better hydrolytic resistance in comparison with the corresponding boronic acids. In addition, these kind of compounds are more soluble in water than original boronic acids. Therefore, several benzoxyborols have been reported as inhibitors of lysyl tRNA synthase for abundant in variety of microorganisms. And this tRNA synthase is an enzyme that belonging to amino acid tRNA synthase family, which are essential for the protein synthesis in all cells. So researchers working on this compounds. I think that was the name of Anacor Pharmaceuticals, and then GSK have discovered a number of benzoxyborole derivatives with the potential application towards the infectional conditions, so as an anti-infective agents. And several other benzoxyborole derivatives have been also designed for the treatment of wide range of disease, including malaria, as I remember, tuberculosis, and some parasitic diseases also. A few benzoboroxol derivatives identified with the improved pharmacokinetic properties and in vivo efficacy studies. This kind of benzoxyborole compounds showed a good efficacy against Plasmodium falciparum and Plasmodium berkei in infected mice. Interestingly, the pharmacokinetic studies revealed that this kind of compounds has metabolic superiority because they have exemplified longer half-life time and then high bioavailability perspectives in dog experiments. Many other biological activities have also been attributed to the benzoxyboroles. For example, as a potent anti-cancer agent, they have IC50 values ranging from 1 to 3 micromolar concentrations against of ovarian carcinoma, breast cancer, and colon cancers. Therefore, we can conclude that benzoxyborole could be used as a promising anti-proliferative and pro-apoptotic compound since it displayed strong cell cycle arresting potency in the phases of G2 and M phases in the cell cycle related to caspase activation in ovarian cancer cell line, thereby leading to cell apoptosis. Therefore, we can use this kind of compounds effectively against ovarian cancer or ovarian cancer therapeutic strategies. The other one, boronic acid and benzoxyborole derivatives. Several other classes of boron compounds have been introduced, including cyclic boronic acids, and then boronic esters, diazoborines, oxazoborines, boronous phosphates, as well as borohydrides. There are too many kind of designed compounds, and then our chemistry team produce in each every day. So, for example, the cyclic boronic acid derivatives were identified as a promising against amyloid beta aggregation because they found that they have ability to inhibit amyloid beta aggregation and provide antioxidant feature, which is useful for the treatment of Alzheimer's disease. So, in work aiming to selectively target proteasomes without inhibiting serine proteases or compound was discovered cyclic boronic acids as potent proteasome inhibitors. The in vitro evaluation, in vitro data revealed that it has an 2 nanomolar concentrations of IC50 values against proteasome activity in colorectal carcinoma cells. So we further evaluated to determine the mechanisms of cell growth inhibition using colorectal carcinoma cells. suggested that proteasome inhibition by cyclic boronic acids induced G2/M, we can say, cell cycle arrest caused an remarkable accumulation of P53, resulting colorectal carcinoma cell apoptotic rates. Then the other kind of important boron-containing compounds are diazoborines, are a family of boron-containing compounds in which the boron atom is stabilized in the form of an aromatic boron-based heterocycle. This boron-containing moiety has been explored as warheads for enzyme inhibition. I want to emphasize that they were discovered as human neutrophil elastase inhibitors. These inhibitors is serine protease and plays important role in several inflammatory disease or conditions because the diazoborine showed IC50 values of 2.7 micromolar against the enzyme, and further studies demonstrated that it has a good stability in human plasma. Oxaborines are another boron-containing class of compounds that have been used in drug design. For example, a series of oxaborines were synthesized and evaluated against NLRP3 inflammasome, the most famous one. This is an important regulator of the sterile inflammatory response, and its dysfunction is implicated in several diseases, including gut or atherosclerosis or type 2 diabetes or Alzheimer's disease. Its activation triggers the intracellular pro-inflammatory response, which includes the release of the pro-inflammatory cytokines like interleukin 1 beta or interleukin 18 levels. So several oxaborine derivatives that were screened displayed promising activity by inhibiting LRP3 inflammasome-dependent release of interleukin 1 beta from macrophages in vitro conditions, and lipopolysaccharide-induced plasma increases in interleukin 1 beta in vivo. So both in vitro findings and in vivo findings collaborated each other. In another findings from the literature, I saw that the boron pyrazole derivative, for example, was found to exhibit antiproliferative activity against hepatocellular carcinoma, another deadly kind of cancers. Further evaluation revealed that oxaboron derivatives acted as an iron chelator, leading to disruption of cell iron uptake and affected the cell signaling pathways of iron regulation. So this mode of action led to S-phase cell cycle arrest and induction of apoptosis in both HEP3B or HEPG2 cells. Interestingly, the pathologic analysis of mice xenografted with triple-negative breast tumor cells indicated these derivatives also induced necrosis, nucleus condensation, deep staining, cell fragmentation, dissolution, and then importantly, neutrophil infiltration in tumor cells. So this is all key findings indicating that oxazoboreans and diazoboreans are potential compounds or their derivatives would be promising against liver carcinoma as well as breast carcinoma via affecting the inflammatory processes, and then cancer progression could be blocked using this strategy. Then another step among the previous studies that contributed to the literature, a study demonstrating the therapeutic efficacy of boron was carried out by our team, starting from our long-standing interest in the search of multi-target directed ligands for the treatment of Alzheimer's disease. We synthesized novel boron-based hybrids structurally different from the available ones. So our structures contained lipoic acid, because lipoic acid was selected as a chemical scaffold, since lipoic acid could easily cross the cellular membrane, especially the blood-brain barrier, and then both oxidized or reduced forms contribute to antioxidant properties of the molecule. So it's a powerful antioxidant since it's a direct scavenger or eliminating reactive oxygen species as well as reactive nitrogen species that are produced following oxidative stress-related age-associated disorders. So taking together all this data, we demonstrated that boron lipoic acid conjugates could be a multi-target compound since it exhibited superior potential in counteracting the neurotoxicity induced by beta amyloid peptides in different neuroblastoma cell line.
Okay.
Ah,
Yeah.
welcome back.
I'm sorry for the connection problem. Finally, let me summarize the last two parts by phone, if you allow me.
Okay.
Okay. The current challenge for the exploration and the application of the potential of boron derivatives to drug design can be divided into three main areas: psychological, logistical, and then insufficient knowledge about the pharmacology, biochemistry, and biology of hybrid organic inorganic compounds containing different boron clusters. The physiological aspects of the challenge of using boron in drug design is seldom raised. This factor has its origins in the fact that since the discovery of boron clusters in the 60s of the last century, their research was the domain of the theorists and then inorganic and materials chemists. Therefore, for decades, this structure, exotic even for organic chemists, did not arouse interest among medicinal chemists and pharmacologists. Therefore, this is another challenge. Finally, as you know, that boron neutron capture therapy has become very famous, especially in the treatment of glioblastoma or prostate cancers. So we are administering borocaptate to the patients. So the huge amounts of these compounds, boron-containing compounds, are being injected to the patients, intravenously injected. So we saw that it is not harmful for the patient. Therefore, the search for boron carriers other than boroseptate or borocaptate has been instrumental in the development of medicinal chemistry clusters. Finally, this is my opinion that the role of boron atom in the new chemical structures is beyond the development of new drugs as it occurred in the past century with chlorine and fluorine addition. Moreover, the recent advances in boron chemistry may result in application in biology and medicine that were recently considered science fiction. For instance, boron-containing compounds could soon be generated to stabilize DNA or RNA structure, heal individuals with the compromising mutations with the substitution of the boron-containing alpha amino acids, participate in smart drug delivery systems, and construct biomaterials with better features than fullerenes and structurally related materials. It is even feasible that boron-containing compounds will eventually be designed to change the appearance of species. That's all. I'm sorry for the connection problems. I want to give my message about the chemistry scientists to develop further boron-containing formulations for humanity. This is my message of the presentation. Thanks for your invitation again.
Thank you, Hassan. Thank you very much for your presentation. And I know your interest in boron-containing compounds and your efforts in the research in this field. So, the last speaker of this morning is Professor Stamatia Vassiliou. Stamatia, good morning.
Good morning.
A few words about Professor Stamatia. She's Associate Professor at the University of Athens, Department of Chemistry. She got her PhD at the University of Athens, Department of Chemistry, Laboratory of Organic Chemistry, discussing a thesis about the synthesis, characterization, and enzymatic studies of metalloenzyme inhibitors. She spent a lot of periods about his postdoctoral research in different universities in France, in Spain, and Germany. Her research interests are focused on medicinal chemistry, especially drug design discovery, synthesis of bioactive compounds, and organic and inorganic chemistry based on organic ligand synthesis for metal complexation. Today, she will speak about this topic, rational development of bacterial ureases inhibitors. Thank you.
So, good morning, and Ivana, I want to thank you for the kind introduction, the invitation to this webinar. I would like to thank you both for the organization of this webinar, and bringing people together is only for good. Even there are technical problems, which I already forgot, but bringing people together, seeing smiles, seeing science, synthetic, medicinal, whatever, is only for good. I really thank you both.
You're welcome. The organizer, so the floor is yours, and then please give us back the organization role, okay?
It's a work, it's a collaboration between the University of Athens and the Wrocław University of Technology. And I will start immediately with the enzyme we studied a long time ago. It's the enzyme urease, and it's an urea amide hydrolase. And the catalytic action of this enzyme is that it transforms urea to ammonia and carbamate, which spontaneously decomposes to another molecule of ammonia and bicarbonate. The enzymatic reaction is 14 orders of magnitude faster than the non-catalyzed one. This enzyme, urease, along with its substrate urea, is related to some very important years in science. I would like to mention just 2 or 3 of them. 1828, it was the first report of urea synthesized by Professor Bolle from inorganic compounds, and this is considered as the birthday of organic chemistry, and I'm an organic chemist, so I have to mention that. Then another important year is 1926, where it was the first time that it was proved that an enzyme is actually a protein. And also 1975, that it was the first time to report a nickel-containing enzyme. For the biological significance of this enzyme, of course, we can understand that it's very important because urea, the substrate, there are a lot of kilos per year extracted by humans each year. It's also the most widely used nitrogen fertilizer, and ammonia coming out from the action of ureas comes back to amino acids by microorganisms. Of course, it's related to pathogenic situations. I would like to mention just two of them: stomach ulcer and soil alkalinization and toxicity, and all of them are related to excess ammonia production that I mentioned at the beginning, that ammonia is the product of urea's action. A special mention to the role of ureas in ulcer. Quite a lot of years before, I don't know about you, but my grandmother, even my mother, used to say that if somebody was suffering from ulcer, you eat spicy foods, you suffer from stress, you eat acidic foods. Maybe they contribute as well, but Professor Marshall and Warren proved that ulcer, it's a question of an infection. It's an infection from the pathogen Helicobacter pylori. This pathogen survives the acidic pH of the stomach because of urea's action, which alkalizes the microenvironment with the ammonia, and thus the pathogen survives and creates ulcer. For this finding, these two professors were awarded the Nobel Prize in Physiology and Medicine in 2005. And the legend says that due to lack of funding, Professor Marshall, in order to prove his theory that the ulcer is a question of infection, he was infected himself by Helicobacter pylori. He treated himself with an antibiotic and he was cured. In this way, he proved that ulcer is a question of infection, not stress, not acidic foods, which of course maybe they contribute, but it's an infection. If we go to the crystal structure of ureas coming from three different sources, like Canavalia ensiformis, Staphylococcus pasteur, and Helicobacter pylori, and we superimpose them, we can see that they are quite similar, and this means that they come from the same ancestor. The role of ureas to ulcer, because it's quite important in my opinion, it's an infection, as I said, and this happens because of ureas. The mechanism is quite important to analyze because it will be used in my presentation, because our inhibitors were based on the mechanism mainly. So I said at the beginning that this is the active site. Urea, the substrate, starts to interact with one nickel ion, and 3 water molecules are expelled. In addition to that, there is a mobile flap opening and closing during the catalytic process, and during this closing, there is an amino acid histidine 323, which approaches, which is not in the catalytic site at the beginning. Urea, the substrate, interacts with other elements of the active site, which make this carbonyl group more nucleophilic, and in this way, the hydroxy group here forms through nucleophilic attack a tetrahedral intermediate. This is very important. Through proton transfer, we move to another intermediate. The mobile flap is opening. Ammonia is coming out and carbamate. Water molecule is entering again, and the catalytic circle is ready to start again. Please keep in mind from this slide the tetrahedral intermediate after the nucleophilic attack, and this histidine 323. They are important for our process. For this slide, just to mention this histidine 323, here you can see in gray the crystal structure of urease without with inhibitor, and in yellow the enzyme in the absence of inhibitor. You see that when there is an inhibitor, the histidine 323 moves a bit closer to the active site. Concerning the inhibitors, there are two main strategies, let's say. The simple one, as you can understand, is creating substrate-like molecules. The substrate is urea. If you construct urea-like molecules, you create inhibitors. One of them is lithostat, which is a marketed drug. The other category is the mechanism-based. According to this strategy, you observe the mechanism, the catalytic mechanism, and according to this, you create structures in order to make inhibitors. And this was the second strategy that we started a long time ago with collaboration Wroclaw. And as I told you, the transition state has a tetrahedral intermediate. So we observed this transition state of the catalytic cycle, and we said, okay, we will make a mimic of it. So in this way, we will make an inhibitor. One of the compounds that are transition state analogs is this organophosphorus compound with 2 nitrogen and 1 oxygen and another oxygen bond. And the crystal structure of this is known, and you can see that the oxygens interact with the 2 nickel ions, which is very important. And based on this and inspired on this, we created some organophosphorus molecules. We avoided phosphorus-nitrogen bonds because phosphorus-nitrogen bond is unstable in acidic pH, and we replaced phosphorus-nitrogen with phosphorus-carbon or phosphorus-oxygen. So we synthesized several analogs, organophosphorus analogs, phosphinic compounds, and among them, we observed one with 620 nanomolar inhibition constant, a phosphinic one, several phosphonic ones, relatively potent. We tried to elongate a bit, and we observed a quite good potency. But the best inhibitor of this series was one where we replaced the oxygen with sulfur. It's a thiophosphinic acid. And we did this replacement. We observed the catalytic activity of 170 nanomolars. Sulfur interacts better with nickel. This is known from inorganic chemistry. And this was the reason we had this good activity. In this study, it was the first time to prove that a phosphinic or a phosphonic compound is indeed a transition state analog, and in this way inhibits the urease. I'm not going into details about the synthesis. It's too much time. Just to mention one thing. In this study, almost each compound had its own synthesis, which is tireful, time-consuming, money-consuming, but of course we had nice results. Just to mention that. With molecular modeling, with 2 of our active compounds, we observed indeed several interactions with the urease active site, but no interaction with the nickel ions. So we decided to make analogs. Compound 1, which was our lead structure from this study, we decided to replace the methyl group here with a hydroxymethyl, and also to elongate from the left part of the molecule. So we synthesized hydroxymethyl compounds, small or long, and in this study, we observed a quite good inhibitory activity, 360 nanomolar. To mention that when we elongated from the left part with ethyl, XC, cyclo-XC, we lost activity almost at all, or we had decreased activity. What this means? This means that urease can accommodate rather small molecules. Again, the synthesis laborious. And with molecular modeling, with 2-hour potent inhibitors, we observed interactions with the active site, but it was the first time to really prove by molecular modeling that these oxygens in the phosphorus atom interact with the 2 nickel ions. This was very important, and it was the first time. And this interaction with the nickel ions, which was not present in the first series I described you, the first round here you can see, was the reason for the improved activity in all cases. If you compare the upper and the lower compounds, which are analogs actually, you see in all cases a better activity, and this can be attributed to the interaction of the oxygen of the phosphinic moiety with the nickel ions, which was our goal as well. Okay. In a previous slide, I told you that we tried to construct elongated analogs in the right part or in the left part. And as I told you, they were not really successful. Actually, they were not inhibitor in most cases. And here is let's say, a dichotomy between what makes an organic chemist happy, I'm an organic chemist, and what makes a medicinal chemist happy. My colleagues in Poland said, okay, nothing. But I was really happy because it was the first time that I used a multi-component reaction. A multi-component reaction is a real tool because you combine up to 6 reagents in one structure, and you can make, by modifying the reagents, a small library or a big library. It was the first time to try a multi-component reaction, and in the organophosphorus chemistry, it's the Kabachnik-Fields 3-component reaction, where you combine an aldehyde, an amine, and a phosphate, and you make alpha amino phosphinic acids just in one reaction. Since then, I really appreciated multi-component reactions, and I use them as much as I can. And there are some of them quite successful. After that, we decide to try covalent inhibition. A few words about covalent inhibition. The traditional reversible inhibition relies on the interaction between the target and the inhibitor in equilibrium. This means there is a binding and unbinding continuously. While in covalent inhibition, you try to make a covalent bond between the target, the protein target, and your inhibitor. This covalent bond is not supposed to happen in the active site, but in the proximity of the active site. And you need also in your inhibitor, the possible inhibitor, a low reactivity group. Just a timeline of covalent inhibitors marketed. I was impressed to know that aspirin is a covalent inhibitor and penicillin is a covalent inhibitor. Just to mention that. Okay, going back to what you need to make a covalent inhibitor, at least to design a covalent inhibitor. You need two parts. The one part comes from the protein part or the enzyme part, and it's nucleophile. The best and the most used is the sulfhydryl group of cysteine. From the other part, from the inhibitor part, you need an electrophile. So nucleophile, electrophile, classical terms for organic chemists. Now it's a classical term for medicinal chemists as well. What are the most used electrophiles for covalent inhibition? Acrylamides by far. Triple bonds, epoxides, whatever, several of them, but acrylamides are by far the most found. And sulfhydryl group of cysteine, again, by far the most used. Now, we were lucky enough, in our case, to have a cysteine in the proximity of the active site. As I told you, we want a cysteine in the proximity, not directly in the active site. And as I explained you, cysteine is not involved in the active site. There is a cysteine in the proximity of the active site. So, okay, we said we are lucky enough to have a cysteine. It's cysteine 322, just close to histidine 322 I mentioned in the mechanism. We know how to make interactions with the active site. We have the knowledge. We were successful in that, how to interact with the two nickel ions. Now we have to add a group to interact with the cysteine 322. So the first idea was to introduce this synamic scaffold onto organophosphorus compounds. Why the synamic? Here, there is a double bond, the one that is good enough for covalent inhibition. I mentioned to you that several types of double bonds are good for covalent inhibition. Here for the organophosphorus part, we made some choices like R group hydrogen, R group hydroxy, R group methyl, hydroxymethyl, aminomethyl. Among them, the most potent was R hydroxy. So in the first set of optimization, we kept hydroxy group and we put several R point substituents here. We had a quite good inhibition for 594 nanomolar inhibition, but if you observe the molecular modeling in the right part of the slide, you see that indeed this compound can interact with the two nickel ions, interact with other aspects of the active site, but there is no covalent bond. Why there was not covalent bond? Because this synamic part, the double bond, was not close enough to sulfhydryl group. It was a little far away, let's say in simple terms. It was not close enough to make the covalent bond happen. So what was our thought in order to optimize our inhibitors? To make a little longer the distance between the organophosphorus compound and the synamic part. So this was the synthesis of the previous, not going into details, I don't have time. Here, in blue, you can see the extension we made so that the double bond comes closer to the sulfhydryl group of cysteine 322. And here we observed a quite good potency, the most potent being the one with 3 carbon atoms and R group hydrogen with 500 almost nanomolar. And with molecular modeling, we were able to show that the covalent bond indeed is happening between the double bond and cysteine 322. Again, the synthesis, not going into details, and we moved forward. In 2017, Professor Curley from the University of Bologna, since we are in Italy, the hosts are in Italy, suggested that catechol, this simple organic molecule, is a covalent inhibitor of urease. And he proved, he had a crystal structure, and he proved that catechol interacts with cysteine 322, makes a covalent bond, and also oxidizes catechol to orthoquinone. And inspired by that, okay, we said, what are we going to do? Simple organic molecules containing catechol. We made several esters. Again, through molecular modeling, we observed the covalent bond formed with cysteine 322, but there were no other interactions with the rest of the active site. Actually, there were very simple structures, not well designed. The most potent of this series was this ester, which contains catechol group and a triple bond. We made several analogs of this, and here I will mention once again what makes an organic chemist happy and what makes a medicinal chemistry happy. During this study, it was the first time to try late-stage fractionalization concept. It's a very useful concept, a very useful strategy. It involves chemoselective transformations at the last step of the synthesis, and the most successful, again, late-stage functionalization reaction is click reaction. Maybe most of you know it, maybe you have used it. I saw it in previous presentations that some of you worked on them. It involves an azide, an alkyne, and copper 1, and you take, at the last step of the synthesis, a triazole. Click reaction was introduced by Professor Barry Sharpless, who took the Nobel Prize in Chemistry last year for this finding. Actually, it's a very chemoselective reaction. And I used it. It was the first time to use this strategy. I had this triazole compounds containing catechol group, but as I told you, they were inactive. So again, once again, happy for the strategy, not good results. This happens quite frequently. Anyway, we moved further. You know, we had the catechol group forming covalent bond with cysteine 322, but we didn't have other interactions. But we have the knowledge. We know how to interact with the active site of the enzyme. How? We introduced organophosphorus moieties. We did it again. So we synthesized several organophosphorus containing compounds with catechol group, all of them. And again, with molecular modeling, we proved the covalent bond between catechol group and cysteine 322 in two cases. Here you can see. The synthesis, again, each compound had its own synthetic pathway. Tremendous work, a lot of work, but very good results to say. And then sometimes, I don't know about you, but it came the time to go back to basics. You remember at the beginning of my presentation, I told you that we have chosen the difficult pathway, the mechanism-based inhibitors. Okay, we had success. Let's go to basic. And why we decided to go to basic? Because in all these studies, what we lack? We lack crystal structure. In all these studies, the proof was with molecular modeling. We tried a lot to have crystal structures, but we were not successful. At that time, the people in the collaborators in Wrocław purchased the cryo-electron microscopy instrument. So we said, okay, we have to try. We have to try again and again. And we said, okay, let's try with something simple. What is the simple? Substrate lack inhibitors. What is the substrate? Urea. Okay, let's not try with urea. Let's try with thiourea, because I told you that sulfur interacts better with nickel ions. Let's make several thiourea containing analogs. We did. We made some of them. And we got 3 quite potent inhibitors. A lot of work again from the students who worked on them. 2 of them, 2 thiosemicarbazones were the most potent with 390 nanomolar inhibitory activity. One thiocarbohydrazone with 223 micromolar inhibitory activity. The modeling. Now we are at the level of modeling. We are at the stage of trying to crystallize the inhibitors with the enzyme. But even though with molecular modeling, we observe the interaction of sulfur with nickel ions in all cases, which proves the correctness of our hypothesis, and several other interactions with other elements of the active site. The synthesis, again, days and days of work from the students. And I told you, where are we moving now? We would like to have a crystal structure. We don't have, it's, let's say, a holy grail. After so many times you try and try, you say, yes, I will manage, maybe not. Anyway, we are at the level of trying to obtain enzyme inhibitor crystal. For the moment, we don't have. But also, we are moving to other pathogenic microorganisms like Cryptococcus neoformans, which is a very pathogenic one. We are working now on that. And also, again, we continue with substrate-like inhibitors. Why not? Finishing my presentation, what else but thanking the people I collaborated so many years, Lucas, Artur, Agnieszka, all of them in Wrocław, in this part of Poland, like the nice collaboration I have with this part of Italy, this small part of Italy, this small part of Wrocław. Nice people, nice collaborations, fruitful results in the end, with a lot of work, And of course, do not forget to thank the students who work on that. And finishing my presentation, again, thank you for this excellent webinar. Thank you again.
Bye. Okay. Nice presentation, Stamattia. Thank you very much. Thank you for your presentation. And great medicinal chemistry approach for the synthesis of your compound. Congratulations. So now we finish and we start again the session in the afternoon at 2:15.